Welcome everybody. Thank you for joining our today's presentation about the endemic Nepalese bat. Please everybody switch off your microphone and your camera during the presentation. After the presentation, feel free to ask questions and we can have a discussion. You can ask your question in the chat room and you can ask the question also personally. And yeah, welcome Sanjeev, and thank you very much for giving this presentation to us today. Sanjeev Baniya is a bat researcher in Nepal and part of REWILD Institute. And today he wants to present the endemic Nepalese bat to us. So thank you very much, Sanjeev, for presenting this topic. And thanks very much to the European Wilderness Society for the opportunity that we can give this presentation and organize it together with you. So thank you very much and looking very much forward to hear about the endemic Nepalese bat today. Thanks, Sanjeev.
Thank you, Ingrid. Hello, everyone, respected researchers, conservationists, and wildlife lovers that are present here. I hope you all are doing good and are healthy during this COVID-19 pandemic. Today, I'll be presenting on the endemic Nepalese bat, its research aspects and conservation needs, and I'll be focusing more on our recent project, which is funded by REWILD Foundation. So, despite being funded, we haven't been able to conduct uh, the fieldworks as extensively as we would have liked because of this pandemic. So it's the presentation is more focused on the framework of the research and what we will be doing in the field and what type of data we want to gather and what type of research questions that we want to answer. So I'm Sanjeev Baniya. I'm currently involved in Nepal Bat Research and Conservation Union, and I'm also one of the members of REWILD Institute student group. This is the outline of my presentation. I'll be starting with bats in general, what type of researches have been conducted in Nepal, what are the research gaps that are present in our region, and I'll be focusing more on our current project, and we'll be concluding with my future plans and preliminary survey results. So bats in general are the second most diverse group in the world after other rodentia. There are more than 1,400 extant bat species in the world currently. This number tends to grow with increasing research efforts. So in the past 15 years, there have been more than 270 new species described. That's the maximum of any mammalian group. Bats are present everywhere in the world except in Antarctica. They are characterized by their sustained true flight. Bats belong to order Chiroptera, which is categorized into two suborders, Yangochiroptera and Enterochiroptera, based on molecular analysis. The Enterochiroptera consists of 6 different families, which includes family Pteropodidae, or the fruit bats, family Rhinolophidae, Rhinopomatidae, Hippocideridae, Megadramatidae, and Crassionicteridae, and rest of the families belong to order suborder Yangochiroptera. Bats are ecologically important. They help in seed dispersal of various pioneer species such as Solanum and Piper, which help in regenerating cleared forests, cleared lands, degraded lands. And they help in pollinating a variety of plants. Some economically important plants include durian fruit in Southeast Asia, a baobab tree in Africa, and agave plant in South America. So this agave plant is used for tequila. So without bats, there would also be no tequila. So bats also help in suppressing crop pests and very importantly, helping farmers and economically helping them by reducing the use of insecticides and pesticides. And the crop pests like cucumber beetles, moths, crickets, and June bugs, which are harmful to to the crops are eaten by bats, and thus suppressing their number. Bats are abundant and are widely spread, and are also susceptible to climatic changes and habitat conversion, and therefore can be used as environmental indicators. Their guano is rich in nitrogen and phosphorus and has been used for fertilizers. Bats roost in a variety of sites that include caves, trees, old buildings, barks, foliages, mines, etc. Caves in general provide relative permanency, protection from predators, and provide microclimatic stability for different physiological activities like hibernation and reproduction. Bats forage in a variety of areas that include forests, edges, and their foraging habits depend. We can use we can study their wing morphology and echolocation characteristics to look at where these species forage. So the species which have short and broad wings would prefer cluttered habitats. The species which have long or narrow wings would prefer open habitats. For example, Nyctelis species. And okay, so here, sorry, here are some illustrations of ecosystem services provided by bats. The first picture is a Cynopterus sphinx, or the short-nosed fruit bat, which is widely distributed in South Asia. It's feeding on a plant, Ficus auriculata, or called kanyo in Nepali. The other second illustration is of a yellow house bat feeding on insects. So we can see how these bats help in different ecosystem services. The cave roosts are used for a variety of purposes like hibernation and maternity. The first picture is one of the sow caves in Pokhara and is also the hibernation site of a widely spread insectivore bat, Apociderus armiger. The second picture is a maternity aggregation of Rhinolophus affinis. That's also in one of the sow caves in Pokhara. So we humans have been interacting with bats in various ways, and I decided to include this slide because many in this pandemic, many of us forget that we have been living together with bats for such a long time and have been wrongly blaming bats for this pandemic. So the first picture, we have been interacting with bats in through cave tourisms. Bats have been residing in our homes, like Scotophilus erie. The third picture is a Nyctelis noctula, which was rescued, and I derived this picture from Facebook. The locals decided to help this bat by feeding cow's milk, so thinking that it had been dehydrated or something. What I would like to suggest is that cow's milk consists of a high ratio of casein protein, which is indigestible. So instead, if you have to rescue some bats, then it's better to use goat's milk, which has very low ratio of casein protein instead of cow's milk. So we told the local people that, and they released the bat that evening. So the last picture is we can't actually just say the species name based on a picture. So it was present in one of the offices in Annapurna Conservation Area project. So that's that. So bats in Nepal, a checklist of 53 species of bats have been documented in Nepal. That was done in 2010. Among all the bats, none of them are protected under Wildlife National Park and Wildlife Conservation Act of 1973. Among these 53 species of bats, two are listed as critically endangered, one is endangered, two are vulnerable, and three are near threatened in the National Red List. You can see the table here of the National Red List and IUCN Red List of these species. Myotis csorbai is critically endangered in the in the National Red List and is data deficient in the IUCN Red List. I have again highlighted Pileator brachypterus here because this species is under continuous revision, and recently in 2017, this species has been uh, the species that is the species that was said to be Philater brachypterus is actually Ipsugo joffrey, which is data deficient in IUCN red list. So it's important that research has been be carried out to actually understand the colony of this species. So bat researchers in Nepal. The documentation in 2010, we have had 19 additional bat species that were listed as data deficient. But since 2010, these are the type of researches that have been carried out, and their focus on the species. So most of the studies have been focused on Indian flying foxes and Hippoceteris and rhinolophids in the cave bats. Um, what I want to depict here is that the detailed studies that include revisiting sites and actually gathering some data that are helpful in conservation have been very rare and must have only focused on Indian flying foxes. Acoustic studies have been carried out in previously in two regions, Kathmandu Valley and Kaligindiki Canyon. We have a call library of bats in Kathmandu Valley. Radio tracking of Rosetus loscanalti was carried out in one of the villages where people hunt bats. So, but that too was kind of that too couldn't get any good data because of the hilly terrain, because of the difficult terrain of in the hilly parts in Nepal. So since 2010, these species have been recorded for the first time in Nepal. Thylonectarus pulvida, or the greater bamboo bat, in 2019, Scotosus dormeri in 2012, Myotis preta, Cynopterus brachiotis, and Nyctellus aviator in 2018. And we have been recording calls of Tararida Myotis, but haven't been able to capture that species. So the baseline is that there has been increment in bat studies, but the conservation issues that these bats face have hardly been addressed, except there has been developed an action plan has been developed in Kathmandu Valley for conservation of these species. We should be the future studies should follow the similar trend trend of studies that have been that has been conducted in the capital region here in Nepal. So the regional concentration of bat space studies, we have been focusing majorly on cities, like major cities like Pokhara and Kathmandu and their outskirts. Studies that have been conducted in other regions of Nepal are highly exploratory studies only and haven't been followed up. Some even in the lowlands, we have been focusing only majorly on Indian flying studies of Indian flying foxes rather than other data deficient species or threatened species. We also lack archaeological studies of these microchiropterans or insect bats, and have only been focused. I've only majorly been focused. I've majorly been focused on Indian flying foxes. This might be because these are easy to observe and study. Maybe that's the reason most researchers have focused on these species. So here's the map of Nepal with the identified caves. And as you can see, most of the identified caves are concentrated to one region and very rarely to other regions. If we take these identified caves as a measure of bat researchers in Nepal, so there are a lot of unexplored areas where where there could be a lot of caves, but the researchers hasn't yet uh, uh, been conducted in those areas. So there are a lot of research gaps in Nepal, and one of the research gap is that we haven't been able to address the conservation issues or even get a basic idea of the ecology of this endemic Nepalese bat, which is listed as critically endangered and data deficient, but the ecological data which us bat researchers should be focusing has been has there hasn't been as much researches. So the research is titled as Acoustic Survey and Monitoring to lay the foundation for conservation of the endemic sorbas mouse-eared bat in Sengja district. This is the steady cave, and all these species that are flying are Minopterus polyginosus, not the Myotis sorbai. And this cave is also important in that it is the maternity roost of these species. So the study species Myotis sorbai is one of 7 species of subfamily Myotini that are found in Nepal. It is endemic to Nepal. There are only 2 endemic mammals in Nepal. The other one is Himalayan field mouse. This species Myotis sorbai was identified as a new species in 1997, and has been separated from Myotis longi. It is related to the Wintoni Capuccini group of subgenus Leuconi. This simply means that these bats are trawlers, and prefer hunting insects above water bodies. And subgenus Leuconi have large hind feet. This species is listed as data deficient and is known only from 2 locations in Nepal, the Kailash cave and the Godavari forest. In Godavari forest, it was misnated while surveying the forest interiors. So the Kailash cave is the only known cave roost of this bat. This bat, although data deficient and critically endangered, isn't yet listed in protected mammals of Nepal. So this is a sorba's mouse-eared bat. It's it has a dull gray ventral pellets and dark blackish brown dorsal pellets, and it is a small form of Myotis longipes and is evidently darker than Myotis longipes, that the Kashmir cave bat that is found in India, Afghanistan, and China. So this is the sorba's mouse-eared bat, Myotis sorbai. So what's the rationale of this study? So besides the bat being endemic and data deficient, we have known this bat from only a single cave roost, and therefore this cave is more susceptible. Therefore, if this cave undergoes disturbances, tourism, etc., and habitat loss, then the bat will be more susceptible to these human intrusions. And there hasn't been any archaeological studies in Nepal, so we are planning to make this a baseline research, so that such that the studies in future can follow similar kinds of acoustic monitoring, acoustic studies, and therefore tackle greater conservation issues. So the locals in around Kailash cave are 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 developing the cave as a touristic site, and it's important that we as researchers tell them tell them the importance, the significance of this cave, and so that efficient conservation as well as local dependency on these caves are well understood. We are planning to build on the Kathmandu call library, and which will also help as a baseline information for future non-invasive bat surveys. So the aims are that we will be monitoring the colony of this bat seasonally, and study its roost use. We will be identifying the foraging habitats and determine relative use by using passive and active acoustic monitoring. We are planning to develop an acoustic call library of Myotis sorbai and its co-inhabiting species. Future plans, some of the plans also include to identify potential hibernation roost sites and prioritize caves in accordance to bat cave vulnerability indices. For conservation and outreach, we are planning to build eco clubs in nearby schools. Awareness programs, posters, brochures, and presentations will be conducted. we are planning to develop a cave and species conservation guideline for effective conservation and management of caves in this region. So the research questions, the blue highlighted group of words are the methods that will be used to answer these questions. So if are there fluctuations in the colony size of Myotis csorbai in Kailash cave in different seasons? What is the cave primarily used for? Is it for hibernation, maternity, or is it intermediate or for daily torpor? We'll be conducting seasonal colony monitoring for that. At what time, hours after sunset, does the activity of endemic and co-inhabitant species peak? Are there differences in activity peaks between nights, between seasons, and among the extant bat species in the locality? So through passive acoustic monitoring, what type of microhabitat does the endemic as well as other bats in the region prefer for foraging and commuting? Is Myotis csorbai only limited to this cave? We'll be conducting active acoustic monitoring and roost surveys around the caves in the region to answer this question. So we'll also be conducting structured questionnaires to to gain more insights on what local people think of bats and this cave. So we'll also be developing echolocation called library of all the inhabiting bat species in that region, so that future monitoring of the species is possible, is non-invasive. So the methods, so the seasonal colony and roost monitoring, these methods will be repeated in each season. So the preliminary survey will be gathering data from papers, media, personal communication, cave explorations, etc. We'll be going to those caves in that region and be measuring their cave characteristics like cave height, length, entrance height, width, compartments, etc. We'll be looking at the threats in the cave surrounding, whether we'll be looking at the cave's accessibility, tourism, land use changes, temples, if there are any bat hunting instances. So and thus then we'll also be surveying bats in the cave and in the nearby surrounding through direct counts, capture studies, photographs, morphometrics, and acoustics. So this is the study area. The center of these two concentric circles is the Kailash cave. So we'll be conducting extensive active acoustic monitoring in the 2 kilometer radii in 2 kilometers concentric circle, and we'll only be looking for caves in the 5 kilometer concentric circles, and we'll be identifying sites for conducting passive acoustic monitoring in 2 kilometer radius as well as in outside 2 kilometer radius. So passive acoustic monitoring, we'll be using audio modes. We'll be using 7 audio modes in 7 sites. The sites will be selected on the basis of microhabitat and land use. The distance between any 2 sites will be made sure that it's greater than 250 meters to prevent any overlap between calls. The sensor will be orientated in such a way that the 2 sensors from 2 detectors are not oriented to the same area. The height from the ground, the height of sensor from the ground will be made sure that it's greater than 2 meters. I'm afraid we cannot account for vertical heterogeneity in our study. We'll be deploying these audio modes for 5 nights in each site, and we'll be recording from 30 minutes before sunset to 30 minutes after sunrise. The covariates in these sites also include nightly temperature, wind speed, distance to nearest water source, distance to nearest clutter, weather, whether it's raining, thunderstorms, moon phase, and cloud cover. So this is the basic plan for passive acoustic monitoring. So it's very important to note the clutter that's around this audio mode, and also account for atmospheric attenuation during our study. This is the audio mode trial. I haven't been able to go to the field, so I've been conducting acoustic surveys in nearby my house recently. So for active acoustic monitoring, we will be using Echometer Touch 2 Pro. It will be used to develop bat call library. Calls of recorded will be capturing bats through misnets, hard traps, hannets, etc. And the calls will be recorded by either by using light tags or making tent houses and letting the bats fly in those houses, and also following the bats after release. So acoustic recording will be conducted at different stress levels. So when bats are tangled in nets, inside bat bag during handling and at release to account for different stress calls too. We will be conducting exploratory and emergence count surveys through the use of this active acoustic monitoring. Exploratory surveys at other identified keep sites will be conducted to make sure to to look if the endemic Nepalese bat is present there. We'll be identifying potential sites for misnating in forested habitat using this active acoustic monitoring. So the data will be analyzed in Kaleidoscope Pro and Raven Pro. Firstly, the noise files will be filtered out using Kaleidoscope Pro. Call parameters will be extracted using Raven Pro. The call parameters include maximum frequency, minimum frequency, bandwidth, peak frequency, call duration, interval, etc. Any calls which cannot be assigned to species level will be categorized at genus level. For example, Pipistrellus coromandra and Pipistrellus javanicus have similar end frequencies with similar type of call structure. So if those species aren't captured, then the two species will be assigned as a single genus in the Pipistrellus genus. A bat pass will be defined as a species of as a series of calls separated from one series of calls by at least 1 second of silence. We'll be using linear discriminant analysis for call separation and thus building a call library. Bat capture will be using hard traps, misnets, and hand nets to capture bats, and the morphometric measurements will be morphometric measurements that include forearm, phalanges, tibia, ear, tragus, and everything that can be measured will be measured. So the statistical analysis of these studies, this is something that's going to improve on with the type of data that I will be gathering from my fieldwork. So the basic idea is that we'll be looking at species assemblages variation during different seasons and among sites. So the, so we'll be using repeated measures ANOVA, or if the data is non-parametric, then Friedman test. We'll be modeling bat passes and feeding buzzes with covariates after exploring data and looking at the data if it follows a certain type of distribution, and we'll be using generalized linear modeling or additive modeling or generalized linear mixed modeling. The activity peaks of species of different of between sites and within and among nights will be calculated through SAT scan statistics. The effects of K microclimate or external temperature on colony size of the bats that are present in these caves will be looked at through correlation coefficients. So for conservation and outreach, we'll be using conservation materials such as posters, brochures, presentations, all in native languages. We are planning to develop a species-specific action plan based on our findings in these regions. We have already identified a citizen scientist from our study area, and he will be helping us with current studies as well as future monitoring. Eco clubs will be made in nearby schools to ensure local level conservation of caves, bats, and the habitat. We are planning to look at the perceptions of local people and tourists on these caves and bats through structured questionnaires. The, the first picture in this slide is in one of the study sites of this research. This study site had been there had been an owl festival, Nepal Owl Festival 2020, occurred in this site. So it was a great opportunity to uh, to um, to gain more knowledge about the study area and about the other caves that are located in that area. So we gain information. We have gained information of 4 additional caves that are located in and around this area. We'll be conducting exploratory studies through active acoustic monitoring in these areas. So the preliminary survey has already been conducted, and these are the outcomes. I'll be presenting detailed outcomes in coming slides. So these are the bullets of these outcomes. So these are the measurements of KAILASH cave. So it's a double-storied cave in which the lower story always has perennial water flow. There's always water flowing in the lower story of this cave, and the lower story is the lower story passes is the area where there is a colony of Myotis csorbai. And the upper compartment, in the upper compartment, there are various other divisions of compartments in the upper half of this cave. And this upper half um, is a site for maternity colony of Miniopterus fuliginosus, as well as other species of family Rhinolophidae and Eposidaridae. So we did count. We had conducted roost count surveys prior to this project too. So in August 2019, before this project was actually funded, we counted more than 7,000 individuals of bats, of which a vast majority were Miniopterus fuliginosus. And the other inhabiting species included Myotis csorbai, Eposidarius armiger, Rhinolophus epinis, Rhinolophus pearsoni, and Rhinolophus pusillus. So we had conducted some acoustic monitoring in that cave, and we encountered the calls of these species. In February 6, 2020, in the same KAILASH cave, however, we only found a single individual of family Rhinolophidae, and that too in deep torpor. We didn't handle the bat because of ethical reasons in February 2020. This could simply mean that the cave is used only seasonally. So it hasn't been used in winter months, but it has been used prior to winter months. So during the summer seasons. So we believe that Myotis csorbai uses this cave roost only uh, only during uh, only during a selected couple of months and not for an year round. So we also identified one other cave at about 250 meters of KAILASH cave, and another cave was also identified in Rangbang, where the Nepal Owl Festival had taken place. That's 4.1 kilometers from KAILASH cave. We conducted dust emergence counts in the mysterious cave, in the KAILASH cave, in the Rangbang cave, and found 24 individuals of Rhinolophus pusillus, 12 individuals of Rhinolophus epinis, and a single Rhinolophus pearsoni in that cave. So these are the pictures of our preliminary study. So the first one is the species in deep torpor. We decided not to handle that bat and then let it be there. We also were looking for any signs of other biodiversity in these caves. So one we found is a species of huntsman spider, and the lower left picture is of another cave that was found in the vicinity of KAILASH cave, and the other one is Rangbang cave, mysterious cave. The mysterious cave is a vertical cave, so it's like 10 feet down, and then only there are horizontal grounds to that cave. So so the so for safety reasons of children of the local village, the cave is surrounded with barbed wires. So we have identified 6 different habitat types to keep 6 audio moths. So these include human settlements, agricultural fields, closed canopy sima forest, intermittent water source, which dries in the winter season, open canopy sima forest, and bamboo aggregations. So one audio moth will be placed at the entrance of the cave to understand the emergence of the bats present in the cave, and how they and when they return, etc. This is the surrounding habitat of KAILASH cave. So these are some acoustic call records that we recorded during our study. So the Ipposideros family Ipposideridae usually has a call structure of constant frequency and frequency modulation. So the Ipposideros armiger has a constant frequency at 70 kilohertz. Ipposideros tenureus has a constant frequency at 153 kilohertz. Ipposideros pomona has has constant frequency at 124 kilohertz. Rhinolophus pearsoni, the call structure of Rhinolophidae is frequency modulation, constant frequency, and then again frequency modulation. So the constant frequency is usually at 60 kilohertz for Rhinolophus pearsoni. For Rhinolophus affinis, it's 88 kilohertz. For Rhinolophus bacillus, this could be possible. The bacillus group is a complex of many other species. So until we have handled the species, we cannot just um, um, be certain about the species. So it's at 105 kilohertz. Myotis csorbai has frequency modulation called structure. So the end frequency lies at 65 kilohertz, and the peak frequency is at 68 kilohertz for Myotis csorbai. The Minipterus polynosus has an end frequency at about 53 kilohertz. So these are the echolocation call characteristics with the mean and standard deviation. We collected this call information through emergence counts, as well as through species while releasing the species after conducting proper morphometrics. So these are this is the table where you can look at this call structure and how the parameters are different from each other in different species. So the limitations of the project is that uh, the activity of bats greatly depend on the abundance of insects. So we would have loved to have insect evidence as a covariate for this study, but due to lack of any insect traps, we I think we will have to assume an equal distribution of insects in these areas. So if we can get insect traps in the future, then great. We will be collecting information on insect abundances in different microhabitats. We don't have enough habitat replicates, spatial replicates, due to limited number of audio moths. There are only 7 in number, and each habitat is replicated only once. So we would have loved to have different we would have loved to have additional number of audio mods to get to replicate this research in different in a proper way, in as many at least for at least at 3 times, at 3 different replicates. So it's already there has already been delay in data collection for the first phase. So we will be conducting the research from the second season, hopefully. We won't be conducting any molecular analysis for species verification. Bat capture survey in all habitats isn't possible due to difficult terrain and limited number of ecotone mismatch. So the future plans include that, as I have already shown in the map of Nepal, that there are about 52 identified caves in Nepal, and the rest of the regions are yet to be discovered, are yet to be studied to get knowledge of the caves in those regions. So the caves that we have already identified, we aim to use bat cave vulnerability index in those caves, and thus prioritize those caves accordingly, in accordance to sub-indices like biotic sub-indices, that is biotic potential and biotic vulnerability. Biotic potential uh, is simply the bat cave biodiversity. So we'll be collecting information about bats, their diversity, their population, if there are any endemic bats in those caves, are there any bats of conservation status. We'll be calculating their rarity. For biotic vulnerability, we will be focusing more on the landscape features and threats of the cave. We will be looking at the accessibility of caves. Are there any the cave openings? Are there any touristic activities taking place in those caves? Are local people using those caves for mining or guano collection, or and land use changes in and around that cave, and thus building on a bat cave vulnerability index, so that it helps the policy makers to make efficient use of funds for conservation of caves and bats in Nepal. So acknowledgement, finally, I'd like to acknowledge Report Small Grants for funding this project, Idea Wild for providing equipment grants, Colonel Slap of Ornithology for giving us access to Raven Pro software, the KAILASH Cave Management Committee, which are very helpful and are optimistic that this research will hold much significance in their livelihood too. Dr. Pushparaj Acharya, Dr. Zoe O'Keefe, and Dr. Gabor csorbai, and Niels Buillard for making this a project, for helping in designing this project, and my field assistants. Thank you. So these are two species, Siposiderus armiger and Rhinolophus affinis. Maybe intraspecific competition inside roost, interspecific competition inside roost. Thank you.
Thank you very much, Sanjeev, for your such interesting presentation. It's amazing what you told us about the bats in Nepal and the endemic Nepalese bat. And I hope that there's going to be a lot more research and conservation in Nepal and in all other countries where so much conservation for bats can be done.